Welcome to the third lecture of second week. This particular lecture we will be looking into techniques we normally use it for diagnosis and many other clinical genetical applications. Basically, we will be looking into the approaches we use for DNA cloning and hybridization. So, we will be covering some of the techniques like southern hybridization, cloning, sequencing, PCR. So, these are the approaches generally being used to understand the genotype. So, when you talk about genotype, we are referring to changes in the DNA, and how would you understand the sequence difference in the DNA. So, you need to clone, make copies of a particular segment of DNA, make a sequence of it and look into what sequence, how it may affect the individual, or because it affects a gene or not. So, these are the approaches people use to understand the difference in the DNA. So, we will be looking into some of these issues, and first let us look into some aspects. So, we in the last week, the last lecture, we looked into pedigrees, wherein we have looked at the phenotype in the individuals of various generations and try to decipher whether the phenotype that you see is because of a defect in a gene, and that has autosomal dominant phenotype, recessive phenotype, or X-linked, Y-linked, and so on. So, to understand how the disease is caused, we need to identify the gene and identify where is the variation. So, this is a very demanding task. So, we will be giving some of the approaches people use to identify the gene defect. So, when you talk about gene defect, the genes are nothing but a small segment of DNA that are present in our chromosomes. So, the cartoon that is shown here represent a chromosome, and you have a long stretch of DNA that comes out, and different segment may have different gene, like for example, the one that is shown here, the arrow indicates the orientation of the gene. If you look more carefully, what is there in that region is that this is a segment of the gene which has got 3 exons, and the 3 exons are regions that are present in the mature RNA. So, when the RNA gets to the cytoplasm, you have the machinery that reads the codes and then translate that code into amino acids. So, that is what is shown here. Some codes have been shown, and which codes for example, phenylalanine, arginine, and so on. Now, what happens in case of disease? So, you could have some change in the DNA sequence, which may affect the way the gene functions, or which may affect the way the protein functions, or the protein may not be made, or made but not properly functioning, and so on. So, what we have shown here is a pine mutation, something that we discussed in our previous class. The pine mutation is you have in a particular base, for example, here the G got transferred to A. As a result, whatever amino acid that should be coded now is not being coded there, but rather the synthesis of protein is stopped because now the TAG represents a stop codon. The question is, how do you really identify? But before we get into that, let us see some of the nomenclatures, or how do we name the mutations. So, this is one of the standard nomenclatures. For example, if you are talking about the DNA sequence, you can see a particular mutant allele is designated with this particular nomenclature here. For example, it is a hypothetical. So, the C stands for coding sequence, because it is present in a coding sequence because it codes for a protein, and the 362 is the number from the first nucleotide of the start codon, the say methionine that coded by ATG. So, in that particular gene, A represent 1, and this particular G which got converted to A represent 362. So, now this is a nomenclature is for the protein, because that is denoted by the letter P, which stands for protein. So, here there is tryptophan, that is what present here, which is the residue 121 residue in the protein, is replaced by a codon for terminating the synthesis, that is stop codon. So, this is how you represent as to what is the mutant allele both at the level of DNA and at the level of protein. Now, let us ask the question, so how do we really know, right? So, how do you know that this is the 362nd residue of a gene, and this is, for example, the amino acid 121st, right. So, normally these informations come from sequencing different segments of the DNA that harbor or that contain the gene sequences, right. So, we derive these sequences from these DNA fragments, and these DNA fragments are derived from a kind of catalog or library we call. One is called as Genomic DNA Library, other one is cDNA library. So, the Genomic DNA Library we will be discussing little late soon. So, this represent almost all the segments of your chromosomal DNA, and we talk about cDNA library that represents your transcribed regions. So, why do you call them as a library? So, what I have shown here is a photograph of the library that is there in our institute. This is the famous Kelkar Library of Indian Institute of Technology, Kanpur, which houses thousands and thousands of books, reference materials, journals, and many other such literature that we normally consult for our research and teaching and so on. And what is interesting is that any library if you get in, you will find racks of books like what is shown here, and they are all stacked there, right. Now, there are thousands and thousands of books. Now, how do you know where is the book that you are looking for? Say I am looking for a book that discusses about human molecular genetics, then I go and search for it. So, how would I really search? You know that in the old one of the traditional way of doing it is that you have such drawers which are attached with a particular alphabet. So, if I book starts with the title human molecular genetics, I will go to a particular rack which has got starts with H, pull out and then look at all the cards that are there, and one particular card would have this particular book name, and it would tell me in which rack, which floor this particular book is there. So, then with that information I go there and pick up. So, otherwise a library is nothing but it is very similar. If you go to a bookshop where they are selling the book, if you want to identify a given book, it is extremely difficult for you to get without anybody's help. So, that is the difference. So, in a library you can go and get whatever you want, although it houses thousands of such literature, books and so on. So, that is something very similar what you see in genomic library or cDNA library. That is why they are called as library. They have everything that you look for, but there is also a way by which you will be able to go and get the particular fragment, right. Let us have a look at it. This is the definition. I quote here from Wikipedia because it is easy for you to go and refer to the Wikipedia. So, if you give a search command called genomic library, it would define it as a genomic library is a collection of total genomic DNA from a single organism. Likewise, if you give a search command for library, it would also tell you that represent collection of all the books that are available, right. So, this is one, it is collection, and second you will be able to retrieve whichever fragment that you want, right. That is this is a way by which we will be able to get. On the other hand, cDNA library is a combination of cloned cDNA. You call it a cDNA because they are complementary DNA, and they are complementary DNA because these are DNA derived from messenger RNA. You copy the messenger RNA which is single stranded, therefore, they are complementary cDNA. The fragments of the cDNA are inserted into again some host cell, likewise for example, in the DNA genomic library, and this cDNA library represent your transcribed regions. For example, majority of the messenger RNA that are expressed in a given tissue if you have developed a cDNA library for example, brain and so on. So, let us have a look at how genomic DNA library is made. This is what it is. You pull, you have the DNA that is running from top to bottom in a chromosome, and what you need to do is normally that you have to cut them into smaller pieces, and not only that, you have to sort them and then store them. And why do you store? for two reasons. One, you do not mix one with the other, therefore, the identity is lost. For example, if I pull out randomly the different pages of different books and bind them together as a book, you will not be able to make any sense of it. So, you need to have them separately. So, that is exactly one of the goals when you make library, that you have certain containers into which you are able to put one particular type, a fragment. It could be multiple copies, but represent this particular region of this chromosome, and then sort them like this, right. And these are smaller DNA fragments, and then you store them, and whenever you want you will be able to get them. So, how do you really do that? So, this is the schematic. So, basically you extract the DNA. So, any of our tissue will give very similar DNA, because there is no big difference between the DNA that is present in my skin as compared to my DNA that is present in my white blood cells. So, I can use any tissue that I think that is available. The easiest thing is that, for example, I can donate some amount of blood, isolate the W with these white blood cells, and then extract the DNA. And then I use these scissors to cut the DNA like what is shown here. And what are these scissors? These are called as restriction enzymes. We will look into that. These are enzymes which are proteins which cut the DNA at specified region, and once you made these kind of different pieces, and you will be able to put them in a specialized host called as vectors that are shown here, and then these vectors get into host organism. For example, here it is bacterial cells, which are like your containers. Now, each one can be put in different container, and we can store them whenever you want. We can pull out one and make as much DNA that you require for any of your downstream applications. So, this process, cutting the DNA is done by a restriction enzyme, and then putting the each fragment, you use specialized host to carry the DNA. These are called the DNA vector. So, these are two important components of a DNA library that we are going to look into. So, what are restriction enzymes? As I said, these are like scissors, they cut. So, what is special about them is that they recognize and cut DNA at a particular sequence, meaning if there are 10 such sites in my DNA, if I add enzyme, it will cut exactly at the 10 such sites. So, suppose for example, I have a DNA sequence that does not have any of that sequence that would be identified by the restriction enzyme, it will not cut no matter how much enzyme I add. So, they are so specific to the sequence, and that specificity really helps us to make the libraries. So, these are proteins as I referred to, and normally these are expressed in bacterial cells, and these are some sort of defense mechanisms for the bacteria to protect from foreign DNA getting into them. So, that is why they make these enzymes, and we identified those genes and we exploited them for our own benefit, that is to make the restriction endonucleases, which we use it for such kind of applications. So, how does it work? So, let us see some cartoon here. So, what is shown here is a DNA segment, and you see a particular sequence which is shown in the red color font, for example, GAATTC. So, this is a sequence that is recognized by an enzyme, restriction enzyme with a name called EcoR1. Let us not worry about the name, but this is a unique name, and that unique name tagged with a unique restriction site. So, this enzyme cuts only this site, it does not cut anywhere else. So, how does it do? You can see that it pretty much breaks the backbone of the DNA exactly between G and A here, likewise at the other strand. So, as a result, one DNA strand, that double strand that you have seen here, is cut into two fragments, right. So, this is how the enzymes cut, right. So, they cut only where such sequence GAATTC is there. Even if one nucleotide is not the same, for example, it is GATTC, the enzyme will not cut. So, it is so specific to the sequence. So, if for example, if I have a DNA sequence and this enzyme has got 3 sites, and no matter how much, if I add multiple copies of the same DNA sequence, if you add enough amount of this particular enzyme, is going to cut all the fragments exactly at the same site. But if I do the same reaction with little amount of enzyme where it is limiting, so what would happen? So here it might randomly cut, because the amount of enzyme is insufficient. At times what happens, ideally you would like that it would cut here and here, but if you give somewhat lesser amount of the enzyme, then what happens, because of the kinetics, it may cut here in this fragment or here in this fragment and so on. So not all the sites are cut if you, for example, reduce the amount of enzyme, or if you reduce the duration of the reaction, if you give it for only short period. So such reactions really help us to get what is called as overlapping fragments. What do they are? What do you mean by overlapping fragments? These are very, very important. I'll explain to you. Let's see this particular figure. What is that? It's a finger, right? It's very obvious to you that it's a finger. But what is not obvious is it an index finger? Is it a middle finger? Is it the ring finger? Is it the small finger? Except, probably thumb, you can think of any finger that it represents. Even if I add one more finger, it would be very difficult for you to tell whether these two fingers have come from the same hand or different hands. So which one is index, which one is middle, so or it is very, very difficult. So because each one, is independent of each other, it looks like from this figure. However, if I show you everything, then you will be able to tell that this is the small finger, this is the index, middle, ring finger and so on, because you have a connection that each one connects to each other, that really helps you to relate and position them in certain order. And that is where the overlapping fragments, even the DNA, helps us to connect two different fragments and tell the relative order. Let us see how it is. For example, here I am taking enzyme 1 and enzyme 2, but the same DNA fragment. Enzyme 1 has got 3 sites as shown here, enzyme 2 has got 2 sites as shown here. So if I do or perform 2 different digestions for the same DNA, you will find that for this it is cut here, for this it is cut at 3 different sites. What is interesting here is that if you look into these 2 fragments which represent the 2 different digests, there are overlaps. For example, this fragment overlaps with this fragment, whereas this fragment overlaps with the same fragment. So this fragment is able to connect these 2 fragments. Likewise, this fragment is able to connect this and so on. So by using these DNA fragments, although they are independent fragments, and by looking into the overlapping segments, I am able to stitch back probably the entire chromosome if I have a way to do such kind of overlapping. So all the genomic libraries that are made for human or any other species have collection of such fragments. There is not only fragments that represent different parts of the genome, but they also have overlaps. That is why I am able to connect one fragment with the other, and I am able to recreate the entire chromosomes. So this is how the restriction enzymes help us to create fragments of our genome, yet we are able to retain some information with regard to the overlap and so on. So that is very, very important in generating DNA libraries. So by doing this, what we do, we have digested the DNA and you have fragments, and we are able to put them in different containers. So what are these containers? I just told you that these are vectors. So what are vectors? There are various types of vectors available. What is being discussed is the most simpler form of vector called as plasmids. These plasmids are circular DNAs that are present naturally in many of the microbes, but the one that we use for a purpose, what is called as a DNA cloning, or the vectors that are man-made. So we have modified a lot, we will come to that a little later. Therefore, they are very, very useful for cloning DNA, meaning to sort the DNA into different containers. So how do you do? This is what it is. So you have a DNA fragment, you have digested them with enzyme which cuts here and here. So I am showing 3 different pieces. What you do, each piece you separate them, and then you use the same enzyme or a combination of enzymes to cut or to make a cut in the circular DNA, and you are able to stick this DNA piece into each one of the plasmids. So, in this way again you have created a circular DNA, but now having a part of the foreign DNA into them, right. How is it possible? It is something like, you are able to cut a rope and then join with a small piece using a glue. For example, well known glue that we use at home is the Fevikwik, very fascinating advertisements. You must have seen that within a second that it's able to stick together. So, very similar kind of glue people use to join DNA, and this glue is called as ligase, and this glue is there in your cell. So, whenever there is a damage in your DNA, and this enzyme is expressed and is able to join, right. So, that is how you are able to maintain your DNA, and such enzymes, which are again proteins, are extracted from many E. coli and other such sources, and we use this protein to stick the two different DNA. Like what, for example, if you have a DNA fragment digested by an enzyme EcoR1 that we discussed earlier, so it would have this kind of what is called as sticky ends, which are complementary to each other. Now, which can now go and form hydrogen bonds. When they do form hydrogen bonds, the this ligase can come and seal it and make it again as a single DNA fragment. So, this is an approach people use to combine two different DNA fragments that were digested with an enzyme, identical enzyme which has overlapping sequence. We are able to recreate the DNA. Again, this ligase is very, very specific. If the sequence that are joining together are not identical, it will not join. If the overhangs are not complementary to each other, it will not join. Therefore, you will not be able to join two different DNA fragments which has sequence difference in the overlapping regions. So, this way we are able to put the foreign DNA inside this circular vector which you call as plasmid. So, this is again a schematic of a plasmid which is used for cloning DNA. And what it has, for example, it has a sequence called ORI, which is the region that is required for replication. So, because the moment this DNA gets into E. coli, it has to make copies, otherwise whenever the E. coli cell divides, this DNA will be lost during cell division. So, it has to make multiple copies. So, this ORI, origin of replication, really helps in making copies of the DNA. Then you have also a region called marker region, called here, for example, is written AMPR, which represent ampicillin resistance, meaning this DNA segment produces a protein which is able to degrade antibiotic, for example, ampicillin. So, this protein would inactivate this antibiotic, therefore, the E. coli can survive if it has got a plasmid. And then you have a long region in which you will be able to clone different fragments like what is shown here. So, that is the region where you can put the foreign DNA. So, this way we are able to pack the vector, and once this vector is ready like what you are shown here, now we can put them inside a host. The host here, what is shown here is a bacterium, E. coli. So, basically you force the DNA to get into the E. coli, and normally the E. coli cell would accept only one such DNA in a cell. So, for example, if you say A, B, C, you see that A plus vector has come here, B has come here, C has come here. So, this is the way they are able to sort them from a complex mixture that you have seen here. Each piece of the DNA we are able to put them inside a plasmid like here, and each plasmid you are able to put inside E. coli. Now, if you culture only this particular E. coli, you are going to make millions of copies of this particular DNA fragment, and that is why it is called as cloning, because we are making multiple copies of the same DNA sequence, right, in a very, very, very cheaper way, because E. coli really does not require any special reagent to make copies of the DNA. So, you give them all the food that it require, they have the machinery to make it. So, this is the way we are able to get a complex DNA that is there present in your cell, extract them and make smaller pieces and put them inside the vectors and then sort them, and then you have cells that are put into containers, right? And then you can go back and get whichever region that you want. That is something that we will discuss a little later. So, this is the process for genomic library. Very similar approach is also used for making a library representing your mRNA, which you call as cDNA library, which represent the transcribed sequences. So, here what we do, your starting material is RNA obviously. So, whichever tissue that you are interested in understanding expression of genes, you take that tissue, extract the RNA, and then the messenger RNAs have unique properties. That is, they have, most of them have what is called as poly A tail, which are polymers of A base, and then we can use this sequence to make what is called as a complementary sequence. So, if you have a primer oligonucleotides that are artificially made, which are complementary to this poly A sequence, and then if you add an enzyme which we described in the central dogma of biology, which is reverse transcriptase, which uses RNA as the template and makes DNA out of it. That is what is shown here. It is able to make a copy of the mRNA. It copies the mRNA to make a DNA, right. So, this DNA can single stranded can be converted into double stranded by a process which we need not get into, but basically what we are trying to say is that we can use the reverse transcriptase to make the complementary DNA, which is cDNA, and then make into double stranded DNA, right. So, this double stranded DNA can now be used to clone into vectors, and that can be made again into library, right. So, that is how we make cDNA libraries, right. So, you have a genomic library that represent different segments of the genome. Likewise, you have cDNA libraries that represent different transcripts that are expressed in a given tissue. You can make one library for brain, one library for liver, one library for kidney, and so on and so forth. That would tell you what are the genes that are expressed. And once you have made the library, the next big task is to sequence them. So, how do we know what are the proteins that are made in our brain? We do not sequence proteins that are extremely complex, difficult to do. What we do is we sequence the RNAs and predict what is called as open reading frames, something that we discussed in the first week lectures, and these open reading frame help us to predict what is the protein sequence that you could make, and you use the protein sequence to predict what could be the function of the protein. So, this is how most of the protein sequence that we know are derived at, right. So, how do you really sequence? So, this is a quick introduction to DNA sequencing, which also uses the same concept that our body uses for making copies of DNA during replication process, right. So, what we have is the DNA segments that are cloned in the vector. So, this is our starting material. Say for example, I am interested in understanding what are the genes that are expressed in brain. So, I pull out a clone from say a cDNA library derived from the brain tissue, and then I try to sequence it. So, what I know in this clone is the vector backbone is common for all the DNA present in that library, right. What is new is the fragments that are present here. So, I do not know the sequence of the fragments, but I do know the sequence of, let us say, the vector that I am starting with. So, what I do is that if I have a vector, and what I do is that I make a small oligonucleotide, meaning these are synthetic DNA which are complementary to the particular DNA region, and as shown here for example, right. And then I denature the DNA, meaning I boil the DNA for example. As a result, all the hydrogen bonds are broken, your DNA is in a single stranded form, and you give very high concentration of this oligonucleotides. They go and bind to the regions that are complementary to the single stranded DNA that is present there, and then you give you add to that tube all these reagents. What are these? These are bases, which are specialized bases. For example, these are not the normal base, these are dideoxy, what you call as, like what is shown here. So, we add the primer we add the DNA templates I already explained, and then of course you have to add the DNA polymerase, which is an enzyme which copies the DNA, and then we add the normal bases that are required for copying the DNA, the deoxy NTPs, and also we add some specialized bases which called dideoxy. So these bases are called as dideoxy because once that base gets added, the the DNA cannot be polymerized any further, meaning the synthesis of a new DNA strand would stop as soon as one of the dideoxy base is added. So that's where we are able to stop. But the trick is that you keep the concentration of dideoxy and deoxy to some ratio such that more often than not your normal base is added. Therefore, the DNA continues to be made, but once in a while your dideoxy base will be added, but if that happens, the synthesis will stop there. So when you do like that kind of a reaction, what you are going to see is something like this. For the same region of the DNA, the primer is bound. For some here, the termination, that is the DNA synthesis stops in the first base, some it stops the second base, some it stops third, fourth, fifth, and so on. So you are going to have different lengths of new DNA that is being made, and what you have done is for each one of the dideoxy bases, you have attached some unique chemicals, which when you excite with certain wavelength of light will give some fluorescence, will emit some light which could be of different wavelength. So by looking at the light, I will be able to call it as base A or base T or base C or base G. So by looking at the fluorescence, I am able to look into and call them as which base it is. So what I do is I separate the DNA fragments based on their size. This is something that we will come back to a little later. So when I separate them depending on their size, the one that are shorter will be here, one that are very long are going to be here, and when I do that, I will be able to call what fluorescence they have, which kind of of the four different fluorescence that I discussed, which are attached to each one of the four bases. I look into what is the fluorescence and call them as a base, something like what is shown here. So what is shown here is the fluorescence intensity. So you see get a green fluorescence, that means I call it as A. If it is red, I call it as T, and then I have stretch of A, and then I get a green, blue, that is C. Again, I get a red, which is T, and so on. So I am able to read the sequence by calling what is the fluorescence that comes out. So this is the way I am able to sequence the DNA. So this is called as one of the robust approach these days people use. This is called as automated DNA sequencing, right? So this way I am able to sequence a fragment of the cDNA, and I am able to get what is the nucleotide sequence, and based on the sequence I will be able to tell what is the protein, right. So this is how these are derived at. So that is how we are able to predict what kind of protein a given gene can code for. We look into the sequence and then we predict the protein sequence. This tells you the protein that a gene can code for. So what we need to do now next is to understand how a change in the DNA sequence can cause a disease, because our course deals with human molecular genetics. So this is something that I am going to discuss in the next class. For this class, we have discussed the basic approaches that people use to generate libraries and then to sequence them, right. So this is what it is, and we will see in the next class.